So before we, I'd like to go over a few logistical details. We'll be taking questions after the presentation, and those who are joining us online are encouraged to type their questions on the chat box. Today's presenter is Seshnee Reddy, the Biobank Coordinator for the National Aquatic Biodiversity Biobank. This role is a collaborative effort between Biodiversity Biobank South Africa and NRF-SAIAB, and it is positioned within the Collections Division platform. Seshnee came on board as Biobank Coordinator in late 2022, although she is already a familiar face with SAIAB from her time completing her honors and MSc degrees at DIFS, Rhodes University, where she also managed the ecophysiology lab. Seshnee played a pivotal role in introducing our biobank during the launch and forum of the Biodiversity Biobanks in Pretoria 2021. Part 2023. So part of her duties includes engaging with both national and international biobanks. So this engagement is crucial, and not only to expand our user base, but also to forge a national strategy for aquatic biodiversity biobanking. Seshnee recently attended and presented at the International Society for Biological Environmental Repositories ISBER conference in Australia, titled Dreaming Beyond Barriers, the Future of Biobanking. So this morning, she will be reflecting on her experience on global biobanking insights, lessons, and perceptions from an international conference and collections. Colleagues, without wasting further time, welcome.
Thank you so much, Nkosinathi, for that awesome introduction. Good morning, everyone, and thank you so much for attending my talk. I really appreciate it. So as Nkosinathi mentioned, at the beginning of this month, I attended an international biobanking conference, which was hosted in Australia, and I also got the opportunity to have a tour of the biobank and collection facility at the Australian Museum. So for today's presentation, I will be giving some insight into my experience, so my perspective on what I learned and what I gained at the conference, as well as visiting a biobank and collection facility in a first world country. So okay, so just to give you a very brief breakdown of the presentation that I'm going to give. So you heard Nkosinathi mention the Biodiversity Biobank South Africa, also known as the BBSA. So I'm not sure if many of you are aware of this network, so I thought I'd just give you a brief breakdown of who they are and how we as the NRF-SAIAB Biobank fit into this network. And then I will give you some insight into the International Society for Biological and Environmental Repositories, which is ISBER, which is the conference that I attended. So I'll just give you a brief breakdown on what that is all about. And then I will give you the presentation that I gave at the conference. So I'm just going to briefly go over some of the slides because some of it, most of you will already be aware of. But if you have any questions that you want to ask me for the ones that I go quickly through, you're more than welcome to ask that during the Q&A session. And then I'll go into the highlights and key takeaways that I got from the conference, as well as visiting the Australian Museum Collection and Biobank. And then we will move on to the fun bit, which are some cool photos from the overall experience. So the Biodiversity Biobank South Africa, or also known as the BBSA, is a research infrastructure project funded by the DSI's South African Research Infrastructure Roadmap. So who are they? Who are the BBSA? So they are a network of repositories of biologically relevant resources. So such as reproductive tissues like seeds, eggs, and sperm, and then also different types of tissues. So like DNA, blood, tissue samples that we store, environmental samples, and microbial cultures. And these samples or resources can be used to support research, capacity development, as well as promote development in agriculture, conservation biology, and environmental management. So then what is the purpose of the BBSA? So the purpose of the BBSA is they are a coordinating network to bring together different biobanks that deal with biodiversity samples. And the main purpose is to increase the range and quality of samples stored in biobanks to improve access for research and development. So some of the BBSA's objectives is to formalize the network. So as I mentioned, to bring together the different biobanks, so existing biobanks within South Africa, to create a collaborative network, and then also to ensure compliance with ethics, legal matters, and permitting, and then to increase the usage of the biobanks. So to promote us, to market the biobanks, to put us out there so that we can increase our user base. And then also for the services of the biobank to be professional and to meet global standards, which is very important. And then also contributing to other related regional, national, and local and international initiatives. So contribute to this and benefit from this. So this is where ISBER will come into play, which is the conference that I attended in Australia. So this is actually the only global platform that addresses technical, scientific, legal, and ethical matters for biobanking. So this is basically like the BBSA, but on a worldwide scale. And then they incorporate all types of biobanks, which is the major difference to the BBSA. So here they incorporate human and medical biobanking and biodiversity biobanks. So the biodiversity biobanks for this conference was actually a recent focus. So in previous years, so this conference is held annually, and in previous years, they didn't really focus on biodiversity biobanks. So this was the first year where they had participants from biodiversity biobanks, so that was pretty cool. So basically in the past, it was mainly focused on human and medical biobanking. So the goals of ISBER, which you will see is pretty similar to the BBSA. So it's to disseminate information on biobanks globally, and then to educate and share information and tools on biobanking, to influence regulation and policy when it comes to biobanking, and to develop best practices guidelines. So ISBER actually has their own publication of best practices, which we have actually been using in the biobank to develop our protocols, and then to provide centralized information on biobanks, and then bringing members of the biobanking community together from different regions of the world. So ISBER 2024, the theme for this was Dreaming Beyond Barriers, the future of biobanking. So the dreaming aspect came about in the Australian culture, the source of all things and the law from which custodianship, mutual respect, and collaboration are grounded. So that's where that came into play. And then we all know that our technologies are constantly evolving. Everything is changing almost every day. So while we are excited for the emerging opportunities, we still have concerns for the uncertainty that they may bring when we try to implement them. So then we have the dreaming beyond these barriers, where we can collectively discuss the obstacles we face, and we can align our practices to mitigate these risks, so we can have more confidence going forward with the evolving technologies. And then ISBER comes into play because through their platform and through the presentation, engagement, activities, and networking that we got the opportunity to do at the conference, we can then start to form and shape the future of biobanking. Obviously, this is not going to happen in one conference, but going forward, eventually, we will come to that aspect of seeing what the future of biobanking will look like. So now I'm just going to go into the presentation that I gave at the conference. I apologize for my Collections and Biobank team because they've seen this before. So the title of the presentation that I gave was South Africa's Hidden Treasure, a National Fish Collection and Linked Aquatic Biobank. I'm not sure if everyone's aware of this, but Nkosinathi did mention that we are named the National Aquatic Biodiversity Biobank by the Department of Science and Innovation. So then obviously everyone knows the location of the biobank here in beautiful Makanda. And then I just gave a brief introduction on the history and development. So in the tissue collection, we have samples that were collected in 1993. Obviously at that stage, we didn't have any infrastructure for this. So the samples were obviously kept in ethanol in the scientists' offices. And then obviously, which is not ideal, but obviously later on, we managed to get -20 household grade freezers and then upgraded to our -80-degree chest freezer. And then eventually we got one -80-degree upright freezer. But then since then, we have obviously increased our infrastructure. So we now have 6 -80 Celsius freezers, which can hold a total capacity of 200,000 samples. And we currently have around 45,000 samples. So we are obviously a growing collections and biobank. Collections and biobanks are always growing. So we have to have the capacity to increase our collections. We also have 2 -40 freezers, which are used for temporary storage of our samples. And then obviously we have our UPS and generator, which everyone knows about. People did mention that overseas, people don't know what load shedding is, but it was an experience having people be like, what is load shedding? And then I had to explain it, and then they were like, why? And then I was like, I don't want to get into it at this point. So our UPS and generators, and then our temperature monitoring system that's monitoring the freezers 24/7. And if something goes wrong, the temperature drops, we get notification so that we can tend to the matter immediately. And then we have a separate sampling space where we do the subsampling of the tissue samples when we have gift requests. And then one of the main things that I sort of focused on, which made our facility unique, is that we also have the wet collection facility. So where we have the voucher specimens. So we have the actual voucher that is linked to the tissue sample, and that actual specimen has been identified by expert taxonomists. So this is quite important that we have the vouchers so that the researchers can go back and they can actually check the voucher specimens. So that, I think, is something that made our facility unique in this aspect. And then obviously I introduced my team. So this was to show everyone that the basic operation of our collection and biobank, so that we have our wet collection, and then we have our data, and then we have our biobanking side. And then I'm not sure whether many of you would know what the scope and objectives are for the biobank. So what is the purpose? What are we aiming to achieve? What are our goals? So the purpose of our biobank is to serve as a repository for the storage, preservation, and management of biological samples derived from aquatic organisms. Then our objectives are to facilitate and support research by providing access to well-curated collections, and then also to promote collaborative research and data sharing, as well as supporting education and outreach so that we can show society why we are relevant. And then all this will lead on to our mission statement, which is to be a hub for research on aquatic taxa in Africa. So this was to show everyone our sample collection and processing procedures, which everyone may already be aware of, but I'll just briefly go over it. So obviously the first step is to get your permits and your ethical clearance. So once that's done and you have all your documents, then we move on to the fieldwork and sample collection. So at our biobank, as you may all know, we have we store genomic DNA and tissue samples of marine, estuarine, and freshwater species, as well as invertebrate and frog species. So images are taken of the specimens, tissue samples are taken, and then the actual voucher is put into formalin. And the tissue samples are then deposited into the biobank, and all the tissue samples have unique labels which will be provided by the biobank. So then the vouchers and your tissue samples are accessioned into the collections. The tissue samples have an ethanol change, so we decant the old ethanol and replace it with fresh ethanol. And then after which all the information that you give us with the tissue samples are then cataloged, images, tissue samples are linked, and then unique locations are automatically generated from our freezers, and then they are moved from short-term storage into long-term storage into the -80 freezers. So then this is a representation of the species that we have from different localities. So the blue dots represent marine species and the green dots represent freshwater species that are collected in South Africa, whereas the gray dots represent all other species that were collected outside of South Africa. And then as you can see, we also have some samples that were collected outside of Africa. So this was just to give an idea of our biobank holdings to showcase what type of samples we have. So obviously, many of you know that the biggest collection that we have are fish. So we have about 189 families of fish. And then we have our sharks and rays. And then we have our frogs and invertebrate species, which are a growing collection. So as you can see for our vertebrates, most of our species were actually collected outside of South Africa, with about 10,055 collected within South Africa, and then majority of our invertebrate species were collected in South Africa. So to give you a representation of the top three freshwater fish families that we have the cyprinids, the cichlids, and the macroclids, with the numbers in black representing the number of species for each of those families. So we have the cichlids, which are the highest at 194, which are followed by the cyprinids at 172. And then our top three marine families are the Serranidae, the carangids, and the sparids, with 77 species of Serranidae, followed by the carangids and the sparids. And then this slide was to give an idea of sample access and distribution, which is quite important, because we have all these samples that I just spoke to you about, but if you need them, how do you access them? What is the procedure of getting hold of those samples if you need them for your research? So all our specimen data is published on an open access and free platform, which is GBIF, the Global Biodiversity Information Facility. And then obviously within SAIAB, you can always come down and speak to us, or you can send us an email. But for people that are outside of the Institute, they can access the information on GBIF, and then they can contact us and let us know, okay, this is a species that I'm working on. I want to know how many samples you have, whether you have these samples from specific locations or anything that they need. And then we will send them a list of those samples along with the material transfer agreement, which many of you will be familiar with. And then they put in their project information on there and get some signatures. And then our curators will have a look at that and they will approve it. Once approved, we will then subsample, and if you're outside of the institute, we will post it to your institute or your university. But obviously, if you are at SAIAB, you just come and collect it from us. So then I went over some of the biobank initiatives and projects that we are currently running. So I think most of you will be aware of the Topotype project, where your morphological reference is your holotype. But in the past, when these holotypes were collected, they didn't have genetic samples. And all those vouchers are now formalized, and it is quite difficult to extract DNA or even high quality DNA from formalized specimens. So our scientists at SAIAB and other scientists are now going out and they are collecting new type material, and all the material is then lodged at the collection and the tissue samples are at the biobank. And then we have the Barcode of Wildlife project, whose main aim is to create a comprehensive reference library of genetic information from endangered species. So we have a list of species. Obviously, SAIAB is leading on the aquatic species. So we go out, collect samples, follow a chain of custody, and then those vouchers and tissues are lodged into our biobank. And then these are used for cases in poaching and trading, illegal trade of wildlife. Then we have a recent project, which is the method development project at the biobank, which was funded by Diplomics. So this is assessment of optimal long and short-term tissue storage protocols yielding maximum high molecular weight genomic DNA for next generation sequencing. So as we all know, that's what we are focusing mainly on now. And for this, you need DNA that's of very high quality. So we are basically, for this, we are testing different preservation methods. So we are testing ethanol versus RNA later versus DESS to see whether these preservation solutions give us higher or lower quality DNA for the long read sequences. So even though they may not necessarily work for the long read sequences, they may still work for the short read sequences, which are for DNA barcoding. And then we have a sample quality and verification project, which is a long-term project, which was funded by the BBSA. So here we basically want to add value to our samples. So we can, if we have a morphological identification, we can verify this with DNA barcoding. And then we also want to work on whole genome sequencing for some of the species for this project. And this will all feed into sample quality management. So we can verify our tissue samples and also DNA extractions to see whether the DNA extracts that we have at the biobank are actually still yielding DNA so that we can actually still use them. So that feeds into a very big project, which basically verifies the quality of all the samples that we have at the biobank. And then to move on to future directions for the biobank. So we obviously want to expand on our current collections, which are fish. So we still want to continue building on that collection. It is a very important collection for us. And then we also want to start securing additional collections. So build on our marine invertebrates and frogs, which is still a growing collection. And then we also want to start keeping different types of extracts. So for example, pharmaceutical extracts from tunicates and marine sponges, which has a lot of value when it comes to drug discovery. And we also want to broaden our user base. So obviously what we focus on at the moment is systematics and taxonomy, which is a very relevant field of study, but we'd like to broaden that into studies such as for climate, environmental, and ecological type studies, because that can increase the user base of the biobank. And then we'd also want to diversify the type of collections that we have. So maybe start collecting and storing eDNA samples and stable isotope samples. And then a very long-term goal of ours will be to start storing viable cloning cells from aquatic species. And you'll see later on when I discuss some of the presentations that were at the conference. As part of the biodiversity section, there was a keynote on cloning where the scientist was prepping to clone the Tasmanian wolf, and he has been using samples, viable cells that were kept at biobanks. So we know it has been done. It's probably way more difficult for fish, but if we have it, then it's available for people to use. So that'll be a very big avenue for the biobank that can send us to the next level. And then obviously my acknowledgements to the Collections and Biobank team who sat with me through a mock presentation and gave me feedback, and also the Fish Systematics and Taxonomy group. And then obviously that was advertising for SAIAB. So if they needed more information, they could come on there and also see the different platforms, not just the collections platform. So then moving on to the highlights and key takeaways from the conference. So obviously my favorite part was the one on the biodiversity biobanks. So it was titled Biobanking in the Natural World, Impact and Offering. So this was actually the symposium that they held on day one. So there was 7 talks. The one that I just mentioned on the Tasmanian wolf was bringing back the past to protect our future. And then I'm biased towards South Africa, obviously. So that was one of my favorite talks, which was by Kim Labuschagne from SANBI, who's a biobank curator there. So it was actually just myself and her that represented South Africa in the biodiversity biobanks symposium at the entire conference. There were, I think, maybe 3 others from South Africa, but they were medical and human biobanking. So that was pretty cool for us. And then another talk that I found pretty interesting was biobanking of marine debris. So the talk was titled When Plastic is Not Fantastic, the Biobanking of Marine Debris as an Emerging Contaminant of Concern. So what they're doing is that they're banking plastic samples. So apparently it will be an easy process. You keep them at room temperature and they just store them in envelopes, or they reuse plastic bags. So the presenter actually said that she brings her empty plastic bags from home, like the bread bags and sweet ones, and obviously they decontaminate it, and then they store the samples in those so that it can be used for research later down the line. And we know that studies to do with plastic and microplastics are increasing now. So that will be, I think, a pretty interesting avenue. I never thought that you could bank plastic samples. So that was pretty cool to see and to hear about. And then So another thing was the human medical biobanking versus the biodiversity biobanks, because I've obviously never been exposed to the human biobanking part of it. So it was cool to see that there were differences, and obviously there were some similarities as well. So obviously now the biodiversity biobanks are being more recognized for their significance to society, because human biobanking, you don't really need to explain yourself much. It's quite obvious why they're important. Whereas I feel like we have to constantly try and explain why we are relevant and why we are important. So one of the cool things that happened is when we were walking around and people saw our tags, the South African Institute for Aquatic Biodiversity, they were like, oh, people were talking about you guys and said that we must keep an eye for the biodiversity biobanks people, because that's going to be something really interesting that's going to happen during the conference. So it was obviously quite new, because they were obviously very used to hearing about the human biobanking and the medical biobanking. So we obviously have some common technologies, like as you can see here, the freezers are pretty similar to what we have. They also have their own database system that they use. And then also when it comes to the ethics and compliance, when the people from the human biobanks didn't think that we have ethics and compliance issues and permits, and we were like, wait a minute, we do. So they just think that, I don't know if they're under the impression that we can just go out and just collect species, you know, as we want. So it was interesting for them to hear that we also have this issue and we hit roadblocks when it comes to ethics and permits. So as part of the conference, I got to visit a biobank in Australia, and this was the Murdoch Children's Research Institute biobank. And that's the actual hospital. It's pretty fancy. And because it's a children's hospital, they actually have an exhibit in the hospital with meerkats. So it's to keep the children calm and, you know, happy when, you know, they're going in there for not so great things. And then this is the database system that they use. So they have over 1 million samples of human tissues and blood samples and saliva. And then that last picture is actually that very fancy automatic freezer that many of you may have heard about. So they have that freezer, and they think it can store probably over 500,000 samples or more. And I realized that because we always wanted something like that, that'll be really cool to have. But in the instance of human biobanking, I think it's more relevant, because the lady that was giving us the talk on this said that they recently got a gift request for tissue samples for 45,000 tissues. So if we had that, we would be going in manually and taking out 45,000 tissue samples and subsampling it. So for them, that they can't do something like that. So this freezer, you just punch in the tissues that you need, and then the machine will go in and select those tissues, put it into a new tray and spit that out for you. So that's pretty cool. Yeah, but it's a very big system. So this is just the front of the system. It runs all the way to the back of the actual biobank facility. So that was very nice to see. Unfortunately, the technicians weren't there, so we couldn't see inside, but they have like a CCTV thing to see inside each of the bays in that freezer. And then I also, as I mentioned, got to visit the biobank and the collections at the Australian Museum. So as you can see, again, pretty similar to what we have, the actual facilities. So they have 11 minus 80-degree freezers, and they have over 100,000 samples. But this is also because they store all samples. So it's not just aquatic. They store mammals, fish, terrestrial. And their biggest collection are tissue samples from whales. And then most of their tissues are obviously collected within Australia and within the Pacific region. So they also have 2 organizations that work with the biobank and the genomics lab facility there, which is the Wildlife Forensics, which is basically the Barcode of Wildlife project. So they get samples in and they have to check what it is, and they use the samples from the biobank as their reference sequence. And then they also have the company Airstrike, which is when planes get birds collide into planes, so they'll get blood samples or feather samples or whatever is left of the bird, they'll send it in here to get sequenced so that they can get a species identification. And then they obviously feed that back to the company so that they can use it for conservation efforts or to potentially change their routes and things like that. So that was pretty interesting as well. And then this is the fish collection, which I got the opportunity to visit. So as you can see, pretty similar to our setup. But what I found cool was that they actually have a larvae collection. Obviously, my background is in larval ecology, so that was pretty interesting. You can see there, that's a little larvae pufferfish, which is pretty cool. And then, so they have, like we have our albino shark and coelacanth. Their main species that they show to visitors when they come in are the goblin sharks, which is really cool. It's just the head of the goblin shark though. And then they also have a juvenile great white shark, which you can see there at the end. So one of the key takeaways that I got from this was, yes, they have a very high number of samples, but the actual facilities are pretty similar to ours. So this is, I was expecting, I don't know what I was expecting, but I expected something that was for some reason way better than what we had, because this is in a first world country and we are in a developing country. But it's pretty cool to see that SAIAB is on par with biobanks and collections like this. And collections are always growing, so we will eventually get to 100,000 samples. So that was really eye-opening and really cool to see that SAIAB is on par with collections and biobanks in first world countries. So moving on to the fun bits with the cool photos. Obviously, wherever we go, we find the fish. So these were cool little metal sculptures that were quite close to the conference venue. And then that little sculpture there at the end was from the Australian Museum. So were the skeletons as well. And they also have a coelacanth at the Australian Museum, which was pretty cool.
Well, isn't that ours?
Maybe it is. I should have asked him. Maybe I should have brought it back with me. And then Australia was very beautiful. Like the buildings and the architecture were all really amazing. I know. I didn't see my charger. So this is Hyde Park. So they had a little fountain there, and that was pretty cool. And then the buildings itself were amazing. We visited some churches as well. This is the inside of one of the churches, and the stained glass windows were very beautiful. And this is the subway station, and then obviously the Sydney Bridge and the Opera House, which was, I never thought I'd get to see that in person. So that was really amazing. And then this is the La Trobe reading room in the State Library. So this was really amazing. So they have different levels where you can view this. And then along here are actually books. So very old archival type books. So it's actually also a collection. So this, the architecture here was amazing. These are actually students sitting there at the reading library. And then we also got to visit, I think it was about 4 streets that had this amazing street art. So that was pretty cool as well. Then yeah, that's it. I'd like to thank you guys for attending. I really appreciate it. And then obviously my funders for the trip, which were the BBSA and SAIAB. And then I'd also like to give a very special thank you to Kim Labuschagne. It was obviously my first time traveling overseas and attending an international conference. So she helped me find my feet, calm me down when I was nervous, when I saw the giant screen that I had to present on for the ISBER conference. So I just want to give her a very special shout out. That is actually a picture of Kim and I that was taken at ACME, which is the Australian Museum for Screen Culture that we managed to visit. So yes, thank you, everyone.
All right, we'll be taking questions. All right, great work done by BBSA and all partners. Is there maybe a spam bank linked to a project? Perhaps with the focus of threatened species on the brink of extinction, maybe? If not in SA, is there any work undertaken outside SA? Nandi ethnic groups.
To be honest, I'm actually not sure about this question, but I can definitely find out and get back to you. As far as I'm aware, SANB does keep live cultures, so that may be somewhere that you can check, but I can definitely get back to you on that. I don't know if Roger or you have any information on that. Okay, cool. We'll get back to you.
Organisation in the UK that I visited, they're a sperm bank, but most people, I think they started off as racehorse
Okay.
sperm banks and then moved into cattle and other farms, and they're beginning to take fish on as well.
Okay.
Can't remember the name of that company. NRF-SAIAB or something.
Yeah, I think you did mention that.
All right, Prof. Michelle Hamer, about how many people were at the conference and what proportion were biodiversity people?
I'm not exactly sure of the exact number. It may have been like probably over 200 maybe. And then it was a very, maybe like 5%, I would say 5 to 10% that were people from biodiversity biobanks or even less.
Okay, Kim saying they keep them at some
there we
level.
go.
Okay.
Thanks, Kim.
Thanks. In the room here? Bongo?
Sorry. Thank you so much for bringing the conference to us. Yes. I love your future directions, especially when I saw the stable isotope component. We are super cool at SAIAB to not have that component. I would really love to see it moving forward. So what you bank at the biobank, do you actually go and sample, or these you also get donations? And if there are donations, is there a specific way where you, a special way or specific way on how to sample what you have to deposit?
Yeah.
And one of the talks that I have with personality, I was just joking and saying I'll deposit my shells there. And then he said, no, you guys damage specimen when you do your sampling. So
Yeah.
I think maybe it would be very cool now that we have you guys here in-house, maybe one day, a workshop or presentation, giving details on how to carefully do the sampling for the biobank or the DNA part of things.
Yeah.
Thanks.
Yeah, thank you, Nakabonga. That's actually a very good suggestion. So we do have protocols in place. We actually don't have protocols in place for marine invertebrates. So that's something that we can perhaps work with your team to do. Unfortunately, with the destructor sampling, we can potentially keep the tissue samples at the biobank, but then to keep the point of the voucher specimen is to have it as in good condition as possible. So if it's damaged, it's not going to be of any use. I mean, you're not going to be able to morphologically identify, you're not going to be able to recheck or do any counts that you need to do. But with the tissue samples, yes, if good pictures, good high quality pictures are taken at the beginning, so before you do your sampling, we can use that. So we'll catalog it into the observation part of the collection. But definitely a good idea to give, to show you guys the SOPs that we have in place for sampling. For donations, sometimes it's not, we don't know that it's coming. They may not know that eventually it's going to come to SAIAB. So we are not fully aware of their sampling procedures, because most of the samples that we get into the biobank are by researchers in SAIAB. So they have our SOPs, they know how to do the sampling and things like that. But it would be good to get it out there on a platform like this, so other people that do eventually want to donate to SAIAB can have an idea of our policies and our procedures.
Yeah, if I can add, so before you can damage the specimen, whatever you're doing, if you can take good quality photographs, because we have an observation collection where we don't, a collection that does not have physical vouchers in the collection, but we keep. So if you can take nice pictures, that collection can be cataloged. Yeah. All right, Paul, great presentation. Thanks, Seshnee. It's good to hear that SAIAB is in par with other institutions globally. Testimony to the staff and all involved. With regards to the, I can't pronounce that. Institute, did you get roughly how much the setup cost? The one that brings straight to you.
For the automatic freezer, I'm actually not sure. Maybe probably in the millions, I would assume, seeing the size of it. But I didn't get the exact cost from the company. But just as I said, that was just the front of it that you could see in the picture. It runs across probably the size of the lecture room at SAIAB. That's how big the back of the system is. So definitely over 1 million, most likely.
All right. Michelle, is it similar to the one that you guys have at Sandy?
Yeah, it is quite similar. So we've got a very small one at Sandby, so it's also a Hamilton. But the one that's at Sandby can only take about probably about 60,000 samples. And that one that they've got, that Seshnee showed, that's the BIOS system. So you can add freezers to it. And so ours is much smaller, but it's the same company, the same, it's also Hamilton. And the one we got cost about 7.5 million rand. And that BIOS one that they've got, that Seshnee showed, is probably about 50 million, just for that size that they've got. So it's very expensive.
No further questions? All right. Just a comment. Francesca, well done, Seshnee. A great presentation and visuals. Great to see that SAIAB is globally leading in biobanking field. To fully match the Aussies, now it's a matter to push for those larvae. All right, watch the space or procedures for collections of samples for stable isotopes. All right. Okay. Kim was asking about, yeah, we lost earlier there for a bit. All right. Questions in the room? All right, that's it. Thank you, Seshnee, for the presentation. See you in the next SAIAB seminar series. Thank you.